All right, we are ready to move on to Chapter 10, Lecture 4. So, in the last lecture, we talked about synapses and crossing over in prophase 1 of meiosis 1. Well, there's something else that happens during prophase 1 of meiosis 1 relating to the meiotic spindle formation and the attachment of the homologous pairs of chromosomes to the meiotic spindle. So remember, in synapsis of prophase 1, homologous pairs of chromosomes are pairing up, right? They're forming those synaptic junctions. Well, not only does that and crossing over happen, but at the same time, the homologous pairs of chromosomes are attaching to the meiotic spindle. But unlike mitosis, where each chromatid is attached to a spindle fiber, in this case, the spindle fibers are attaching to the centromere. Remember, when chromosomes are in the pair of sister chromatid forms, they have a common centromere. So in meiosis, the spindle fibers are attaching to the centromere of each chromosome in the homologous pair. Okay, so what that means is that by the time we get to metaphase 1 of meiosis 1, and I'm not going to worry about switching colors right now. Sneeze. Sorry, you can bless me on your videos while you're listening. Okay, so by the time we get to metaphase 1, we're going to have chromosomes lined up on the metaphase plate, but they're going to be those homologous pairs of chromosomes. So we're going to have basically a double row of chromosomes lined up on the metaphase plate attached to the spindle. Okay, now why is that important? It's important because it is the job of meiosis 1 to separate the homologous pairs of chromosomes into the daughter cells at the end of meiosis 1. Okay, so when this parent cell actually undergoes cytokinesis, we're going to be separating out those homologous pairs of chromosomes from one another. So it's important that each chromosome is attached to a spindle fiber, and then we'll talk a little bit more about what happens next later. Okay, well, the next interesting and important thing that happens during meiosis 1 occurs during metaphase 1, and this is called random alignment. Okay, so first of all, I want us to look at this cell over here where it says possibility 1, and I want us to figure out what is the diploid number for that cell. So remember, the diploid number is when a cell has 2 copies of every chromosome, and because meiosis starts with a diploid germline cell, and by the time we get to metaphase 1, we haven't divided the cells or anything. These cells are still diploid. And the other thing I want you to remember is that you count chromosomes by counting centromeres. So yes, this is post DNA replication that happened in S phase, but each of these chromosomes is in the pair of sister chromatids form, okay? So we've got 1, 2, 3, 4 centromeres. Therefore, we have 4 chromosomes. So our diploid number is 4. All right. Well, so one more thing I want to point out Remember I told you that when synapsis happens, the chromosomes aren't side by side like they're drawn in these pictures. They're actually sandwiched on top of each other. So when the chromosomes are attached to the meiotic spindle, they actually do look more like this because they're attached to the meiotic spindle right there at the centromere. And remember, they were still attached to each other at those synaptic complexes. So they're being pulled apart, and it makes them look almost like a set of double parentheses or something. Okay, well, when the homologous pairs of chromosomes line up on the metaphase plate during meiosis 1, the arrangement of one pair of chromosomes is independent from the arrangement of any other pair of chromosomes. So if we, hypothetically think about our metaphase plate being right down the center of this cell, this is where cytokinesis will happen later, okay? And so we've got the left-hand side and the right-hand side in the way this is drawn, of our metaphase plate. Well, when pair number 1 over here on possibility number 1, when this top pair of chromosomes lines up, it can line up with the chromosome that originally came from dad on the left and the chromosome that originally came from mom on the right. Or, as it's shown over here in possibility 2, if we look at the second pair of chromosomes, they could also line up with the chromosome from dad on the left and the chromosome from mom on the right. But if we look over here at possibility 2, the chromosomes could line up so that one chromosome from dad is on the left from one pair and the other chromosome from dad is on the right in the other pair. Okay? Both of those possibilities are equally likely. These two homologous pairs of chromosomes are completely independent from each other when they are lining up on the metaphase plate. There's no overall organizing pattern that determines whether or not the chromosome that originally came from dad in one pair is on one side of the metaphase plate and the chromosomes that originally came from mom in the other pair is on the other side of the metaphase plate. Okay, there's no organizing pattern to that. It's completely random, which is why we call this random alignment. Okay, well, random alignment happens in metaphase 1. But that leads to independent assortment in anaphase 2. Okay? All right, so at the end of metaphase 1, we've got the chromosomes lined up randomly on the metaphase plate. Okay? Well, at the end of meiosis 1, we're going to have daughter cells that are now haploid. Okay, so remember, our diploid number was 2. After meiosis 1, each of these daughter cells only has 2 chromosomes. So now they are haploid. It's just that the chromosomes are still in the pair of sister chromatids form. Okay? Well, when you look at these daughter cells, now we're in metaphase 2 because the chromosomes are lined up on the metaphase plate. The way in which these chromosomes are arranged in these daughter cells depends on how they lined up on the metaphase plate in metaphase 1. Okay, so this daughter cell number 1, came from possibility number 1 because both chromosomes from dad lined up on the same side of the metaphase plate. So when this cell at the end of meiosis 1 divides, both of these chromosomes wind up in the same cell together. Okay, the 2 red chromosomes wind up in the same cell together. But over here, we had a blue and a red on the same side of the chromosome, and then a red and a blue on the same side of the on the same side of the metaphase plate, excuse me. So when this cell divides, our 2 cells in meiosis 2 have that same arrangement of chromosomes. And remember, all of these possibilities are equally likely, okay? It's completely random. And so at the end of meiosis 2, when we separate out the sister chromatids, we wind up with 4 haploid daughter cells. And each one of these combinations, okay, when possibility number 1 over here on the left, when it separates out its chromatids, it's going to form 2 gametes, okay? And these gametes form combination number 1. They're more or less identical, even though they're really not because of crossing over, okay? But they both have the big chromosome from dad and the little chromosome from dad in the same cell. Okay, so that's combination 1. So each of these combinations, combination 1, 2, 3, and 4, are genetically different from one another. And so we say that in anaphase 2, when the sister chromatids separate, in anaphase 2, they sort independently into the gametes. Okay, so how these top chromosomes sorted into the gametes was independent of how these bottom chromosomes sorted into the gametes, okay? Because of how they lined up on the metaphase plate of metaphase 1, okay? So random alignment leads to independent assortment. An independent assortment increases genetic variability, okay, by a lot, all right? So in our example that we've been working with, our diploid number was 4. Our haploid number was 2. Well, there's a mathematical equation that tells us how many of these possible combinations for gametes can exist for an organism based on their diploid and haploid numbers, okay? Well, when our diploid number was 4, we had 4 possibilities, 4 different combinations for how those chromosomes could sort into the gametes. But the number of possibilities is not equal to the diploid number. The number of possibilities is equal to 2 raised to the haploid number. Okay? Well, in our example, 2 squared is 4. But think about human cells. Okay? Humans, our diploid number is 46. Our haploid number is 23. You take 2 and you raise it to the 23rd power. That is not equal to, that is greater than 8 million possible combinations. So instead of having combination 1, 2, 3, and 4, we'd have combination 1 all the way down to combination 8.4 million something. Possibilities for how the human chromosomes can sort into the gametes. Okay? And then and those 8 million possible combinations are for every time a human germline cell undergoes meiosis. So every time a man makes sperm, there are those over 8 million possible combinations. Okay? So one round of meiosis, you're going to get 4 sperm cells. Well, 4 sperm cells is not very many. Okay? And so men make thousands of sperm cells. Each of those individual cells was a result of one of those 8 million possible combinations. So what are the likelihood that any 2 sperm cells are going to be completely identical? Very, very rare. Okay? And so which of these sperm cells and egg cells happen to come together to fertilize each other is another way that genetic variability increases in organisms that reproduce sexually. Okay, so we've talked about 3 different ways 2 ways in meiosis and 1 way resulting from meiosis that genetic variability in sexually reproducing populations increases. One of them is crossing over that happens in prophase 1 and meiosis 1. The second one is independent assortment. The third one is fertilization, random fertilization. Okay, so there's lots of things going on in meiosis and as a result of meiosis that increases genetic variability in sexually reproducing populations. Why is that genetic variability important? Because genetic variability is required in order for populations of organisms to evolve. Okay, for allele frequencies to change, there has to be